All right, we've finished our look at embryology, and as we go from embryology into our system studies, before we do that, we're going to do a little bit more with kind of the contact zone, that intermediate zone between embryology and then larger anatomy. We're going to talk a little bit about histology. Histology is generally thought of as the study of tissues. Specifically, you're looking at all the different cell types and cell shapes that make up your different tissues, how they're arranged, your cellular arrangement. Are they packed tight together? Are they in rows? Or how are they joined together? The connections between cells. About extracellular materials. So in your bone, for example, most of your bone is not made up of cells. There are cells scattered through your bone, but most of the bone is extracellular material, that hard material of the bone. The same would be true for a cartilage. For blood, for example, it's mostly fluid in there with cells in between. We can think of it as the microscopic study of organs and tissues, what things look like under the microscope. And when you do that, when you look at these organs and tissues under the microscope, you're looking at these cellular arrangements, what kinds of cells, shapes of cells, and so on, or extracellular materials make up those structures, the organs and tissues of the body. The next concept I want to introduce is cell lineage. Cell lineage. So we start with the zygote. Our first cell, it divides, we get more and more cells. As we get more cells, we eventually get the germ layers to form, the endoderm, ectoderm, and mesoderm. And then those regions form more specific types of tissues. So you have the regional specializations. So cells in one part are becoming one kind of structure, and cells in another part become another type of structure, and so on. So, for example, here we have a cell of the ectoderm. As the cells divide, some of them become identifiable as surface ectoderm cells, as opposed to extraembryonic membrane or neural ectoderm or neural crest. So we have surface ectoderm. Some of those cells that are forming the surface ectoderm may eventually form into hair follicles. In there, they fold in and go deep into the dermis. Some of those down in there might form the hair matrix. Hair matrix cells divide and make more hair matrix cells, and some of those matrix cells will eventually form the keratinized cells that make up your hair and inner root sheath of your hair from your hair matrix. So things become more and more specialized. These are referred to as determinate steps. Their fate is more and more determined. So there are genes that are turned off or turned on during these cell division processes in each of these determinate steps. So some genetic change here caused that surface ectoderm to become part of a hair follicle. The hairs, these cells of the ectoderm started to change in such a way that they became, started the root of the hair follicle to turn into the dermis, and then some change within some of those cells caused them to become hair matrix cells. Some genetic change, a determinate step, a gene turned off or turned on to facilitate the behavior of that cell in that tissue. Most of these determinate steps are one way. There's no turning back. So once you've made the change, you can't go back the other way. It would be hard to take a hair matrix cell and then stick it into a muscle and assume that it would function as a muscle cell. You can't put it in the blood and think it's going to act like a blood cell. Once they go to the next stage, the next determinate step, their fate is more and more locked in. They can't go back the other way. The next important concept coming from the idea of cell lineages and how that cells become more and more specific. These terms, totipotent and pluripotent, we'll often run into with regard to what we call stem cells. Cells that are dividing and can have a number of fates down the line. If a cell is totipotent, that means that all of the different cell types of the organism can be derived from that cell. The zygote is considered the ultimate stem cell. As the zygote divides and becomes like 2-cell, 4-cell, 8-cell, some of those cells that divide from that original zygote would also be totipotent. You can grow a whole new organism from some of those individual cells. You can isolate them, they start to divide and eventually form an entire organism. So those cells are totipotent. But fairly rapidly in the early stages of division of the zygote, you're going to get some specialization of the cells. Some of the genes will be turned off. They will no longer have the ability to become all types of cells in the organism. These are referred to as pluripotent cells. Then, if they can't become anything, they might not be able to become anything that's associated with the nervous system, for example, at a certain point in the developmental sequence, or they can't become a gut cell in the developmental sequence. So these are still stem cells. They have a lot of different cell types that might be able to be derived from them. That's where we get the plural, and many different things. But they have a limited potential. They can't become any type of cell in the body. Pluripotent cells. And then eventually these cells become more and more specific and finally become what we call fully differentiated cells. Fully differentiated cells, they can't change to become something else. Sometimes these are referred to as terminal because they no longer divide any further. So they reach a point where they are the last cell in their lineage. They will not undergo another division to form more cells of the same type. So we have some of our body cells are terminal cells. Many of our neurons, our muscle cells, and so on are terminal cells. And then there are some that can produce more cells of the same type. They still are fully differentiated cells, but they will produce more cells of, say, skin cells, lining of your oral cavity, places where you see a turnover in cells frequently in some connective tissues and so on. These are fully differentiated cells, but they still are undergoing cell divisions to make more of the same cell type. So these are different stages, totipotent or pluripotent or fully differentiated. Now that we have a little bit of background on the development of the different tissues, that specialization process whereby we get these specific cell types, let's talk about our functional classifications, our groupings of tissues. There are 4 primary categories. First of all, the epithelial tissues. When we think of epithelium, we think of our skin first off. So we know that's a kind of covering. So in a definition of epithelium, we would talk about coverings and linings. So they would line your oral cavity, your nasal cavity, and all through your gut, inside of your blood vessels, and all those things. So forms coverings and linings. In addition to that, to be an epithelial tissue, it also has to rest on a basement membrane. Rests on a basement membrane. We'll talk about that basement membrane here a little bit later. It's an extracellular layer. It's not made out of cells, not made out of the actual cell material, but something secreted by the cells. It forms a basement membrane. It's kind of a glue that holds the epithelium in place. And usually, because it's a covering or lining, this is associated with the free surface. A free surface. So of course your skin has a free surface, but any other cavity in your body would have a free surface side to the epithelium and then a basement membrane side to the epithelium. So this kind of sets up a polarity in any epithelium, a basal portion of the epithelium and a free surface layer of the epithelium. So anyway, so these are epithelial tissues. Occasionally, a tissue that begins to form as an epithelial layer as part of one of these coverings or linings will migrate away from the surface and embed itself deeper in the tissues where it's no longer associated with a free surface. And it's no longer part of a covering or lining, so it's a dissociated type of epithelium. In that case, we call them epithelioid tissues. They had an epithelial origin. They were part of an epithelium, no longer associated with the free surface. So that sets our epithelioid tissues apart from some of our other tissues here. So portions of your adrenal medulla, for example, would be your pituitary gland is a really good example of an epithelioid tissue. It was at one time part of the roof of your nasal passages in there in the oronasal cavity, and then it migrated away from that position. So there are a few organs in the body that are epithelioid. Started as epithelium, but then moved away from that. We will break down the specific types of epithelial tissues here in just a little bit. The second group are the connective tissues. The connective tissues usually have some kind of a function to join two parts together. That's one possible role. Join structures together. Sometimes they simply fill space. Between structures, they just kind of take up the space in there, so they can be just space fillers, and sometimes they give an overall support or shape to a structure or organ. Very important, of course, in your skeletal system and cartilages and so forth. We have that support mechanism in there, joining bone to muscle. We need that strong support in there. So anyway, these are connective tissues. Included in here, not doing any of those things, but for a number of reasons, blood is included with your connective tissues. Okay? And later on we'll break down all the different types of connective tissues. Now on to our next. Muscle tissues. This is fairly simple to think about what muscle tissue is. We know it's in control of movement. Both our external bodily movement, moving our arms around, our facial expression, doing those kinds of physical movements. So it's responsible for movement. So it's both external, what we've just described as mainly externally visible types of movements, but also internal types of movements, things going on inside your body. It includes all sorts of different things to have cells contract that help to move sweat to the surface of your skin. Cells that contract to cause peristaltic movements through a tube of the reproductive system, the heart, and so on. There are three kinds of muscle tissues. We're going to break them down later on, but I'll go ahead and list them briefly here. What we call skeletal muscle accounts for our voluntary movements, and most of everything connected to the skeleton is part of our voluntary movement control system. So skeletal muscle. And then cardiac muscle, only found in the heart. Cardiac muscle, the heart muscle. And then the remainder of the muscle is what we call smooth muscle. It's found in most of your hollow visceral organs. Those are the organs inside your body that have an empty space in them, some kind of a tubular passage, or like your urinary bladder, a cavity on the inside. So in your visceral organs, also along your blood vessels, you have smooth muscle controlling the diameter of your blood vessels that the blood passes through. Also inside your eye, the focusing mechanism for your eye and the pupil dilation. That's all controlled by a smooth muscle. And then finally, in your skin, there's what we call arrector pili muscles that cause your hairs to stand on end. Your goosebump muscles in your skin are smooth muscles. None of those are voluntary things. We don't have any voluntary control over those. No voluntary control over our heart muscle either. So skeletal muscle, cardiac muscle, and smooth muscle, we'll look at each of those in detail later on. Our fourth tissue category, and this is the last of our tissue categories. We've been through epithelium, connective tissue, and muscle. Nervous tissue is the fourth category. Nervous tissue includes a number of cell types, not all of which are conducting nervous impulses. We kind of connect everything that is associated with the nervous impulse conduction in this category, so it's easy to define what is a nervous tissue, but it includes more than what you might think. So these are associated with neural impulse conduction. Now, when we studied embryology, we saw the development of the neural tube. So everything derived from neural tube is going to be considered a nervous tissue. Also, some of the tissues that come from neural crest, so some neural crest tissues are also included in this category if they are part of a neural impulse conduction structure. So things included in this include neurons. Those are the things in the central nervous system that actually convey the neural impulse. Then associated with neurons, we have a number of what we call neuroglial cells. That are there to support the neurons. And we're going to list those later on. It includes astrocytes, oligodendrocytes, microglial cells, and the ependymal cells. So 4 different categories of neuroglial cells there. Also in our peripheral nervous system, we have something similar to the neuron, and sometimes they're referred to as neurons. More technically, they're referred to as ganglion cells. The ganglion cells. So they function like neurons in the peripheral nervous system. And surrounding some of the ganglion cells, we have what are called satellite cells. Satellite cells, they kind of pack in around and protect the ganglion cells, satellite cells. And we have the Schwann cells that form around the outside of nerve fibers. And we'll describe how that works later on as well. So this kind of finishes out our different tissue categories. We've seen epithelium, connective tissue, muscle tissue, and nervous tissue. Nervous tissue, things associated with impulse conduction derived from either the neural tube or the neural crest.